So I am a neonatologist, but I am the first director of NICHD in 30 years to have actually run a laboratory. And that's a big difference because I have felt your pain. I have submitted many grant applications. Some were funded, some were not funded. But I think I bring a very different perspective to the position. And I was asked to talk about my science today, and I thought that there's no better way to communicate my commitment to model organisms than to show you the research that we are doing in our laboratory that uses the mouse, a model organism. So that's what I'm going to mainly focus on. 90% of my day, which is hardly a 100% effort, it's more like a 400% effort, is related to running NICHD. But fortunately, about half of the NIH Institute directors have their own laboratories. So my laboratory is in the Human Genome Research Institute. Because of conflict of interest regulations, we need to have our lab in another institute, but as a medical geneticist, it's just a perfect opportunity. So NICHD, just very briefly, is undergoing a strategic planning process. I will be available after this talk and also during the lunchtime to answer questions about our strategic planning process. I can assure you that theme 1 is called Understanding Human Development, and we have heard the many comments that were in response to our request for information. We got nearly 1,000 comments, and we heard very clearly about the need to articulate our commitment to model organisms. So I think you will be pleased when you see that. The official strategic plan will be publicly available at the time of our council, which is in early to mid-September. And then you can start talking with your program officers about what that means for your ideas. But again, early human development, development using model organisms is very clearly articulated. So I don't think we should have concerns about that. Okay, green. Let's try this. Okay, is there some trick to... I have to use, okay, I have to use this?
Use these for forwarding.
Okay, I tried that, so I think I figured out how to do it, yeah. So our laboratory, part of our laboratory focuses on developing a prenatal treatment for Down syndrome. And our hypothesis is that prenatal treatment given to the pregnant woman will result in improved fetal brain growth, connectivity, and ultimately improved neurocognition. And the reason why we feel pretty strongly, let's see, does the pointer work? Yes. We feel pretty strongly that treatment should be given prenatally is so much of the difference in brain development between the euploid fetus and the human fetus with Down syndrome. Those differences are occurring in the middle part of the pregnancy. Here's where you can do prenatal testing to confirm the diagnosis. You could start the treatment here, and then the goal would be to improve neurocognition with treatment given to the pregnant woman. Now, why Down syndrome? Well, when I was a medical student, now it seems to be working. When I was a medical student, my mentor at Stanford University School of Medicine, Leonard Herzenberg, had a son, Michael, who had Down syndrome. And he initially charged us with developing a non-invasive prenatal test for Down syndrome. I'm pleased to say that we accomplished that goal. I'm one of the very fortunate group of people who was able to see going from looking under the microscope at intact cells to advancement in technologies that ultimately resulted in cell-free DNA testing in pregnant women. And that is now part of routine prenatal care. Over 10 million genetic tests have been performed, genomic tests, that involve some aspect of DNA sequencing to screen and diagnose for Down syndrome. And the point is that type of testing is done quite early in gestation, so it would be feasible to treat the fetus. And the other advantage is that the developmental anomalies in the brains of human fetuses with Down syndrome are fairly homogeneous. However, there's some significant challenges. So you have this disconnect between very promising studies that have been performed in mice, over 30 of them have been done in 26 completed human trials, but these tests have been very positive in animal models, but they haven't translated to human clinical improvement. Now, the challenges have included that none of these trials has been very big. The maximum number is 350. There's the potential for inter-individual variability. There's been a lack of standardized outcome measures or harmonized efforts at expanding and including and connecting the databases. There's the potential for placebo effects, particularly in a population of people with developmental disabilities. And all the studies have been done fairly late in life because of concerns over safety. But as I showed you, some of the differences are already there at the time of birth. Now, we also have to be concerned with safety, and we have two patients. We have the mother and we have the fetus, and we can't harm either of them, and we cannot increase the number of adverse effects in the developing fetus. And we also have to cross two barriers. We have to cross the placental barrier as well as the blood-brain barrier in the fetus. And we also have the challenge that pregnant women are already self-medicating. They're using the internet to find out about things that may or may not help their fetus once they have a diagnosis of Down syndrome and they're continuing their pregnancy. Okay. So we have worked for a number of years, and if you're interested in the human fetal cell-free transcriptome, there's a large amount of cell-free RNA that is floating in the amniotic fluid surrounding the human fetus. There are still a number of amniocenteses that are being performed to confirm diagnoses in the human fetus, and that amniotic fluid is available once the clinical testing has been done. That RNA is nearly 100% fetal, and it provides a real-time window into early human fetal development. And you also have the karyotype confirmation. So we know that when the fetus has Down syndrome, we can be absolutely sure that we're looking at dysregulated genes. And in comparison to the previous talk, we are not dealing with one gene or one mutation. We're dealing with hundreds of genes in the critical region on human chromosome 21, and those genes have downstream effects on other genes across the human genome. So it's a very, very complex human condition to try to study and treat. However, we found that in the amniotic fluid, there's a significant number of transcripts from the human fetal brain and central nervous system. And furthermore, and all these data are publicly available and are summarized in this paper that I wrote with one of our postdoctoral fellows, Lillian Zwiemer. Each human cytogenetic abnormality, so these are the three most common autosomal anomalies in the human, each one has a distinct signature in terms of pathway dysregulation and gene dysregulation. So the characteristic findings in Down syndrome are oxidative stress and abnormalities in cell proliferation, as well as neuroinflammation. So we hypothesized that if we could treat these pathway abnormalities, that this would hopefully result in really an improvement in the ability to live an independent life. We didn't think that we would be able to change the physical appearance of people with Down syndrome. Our goal is really a more achievable one, which is to increase IQ points by 10 to 20 points to be able to enhance independent living. And I just want to point out that trisomy 18 and Turner syndrome have very different abnormalities, very different pathways are involved, but many of them are quite related to some of the problems that affected individuals have as they age. So older women with Turner syndrome, for example, have significant problems with cholesterol, and they have significant problems with coarctation of the aorta, and we think that we see this even in fetal life. So the approach that I'm going to tell you about right now with Down syndrome is equally feasible for Turner syndrome, for example. Now I realize that this is a bit of a busy slide, but it shows you our overall goal, which is to combine, I see we have in vitro, and the letters are reversed, but basically our goal ultimately is to get back to the human and to be able to do a prenatal clinical trial. But we have both cellular experiments that are being done in induced pluripotent stem cells. We are validating drugs and screening drugs, and then we are also looking at the different animal models of Down syndrome and characterizing them from a phenotypic perspective, and then testing some of the candidate drugs. So the first thing we had to do was figure out which of the 3 most commonly used mouse models of Down syndrome would be appropriate with a prenatal phenotype, and then determine the best endpoints to evaluate therapy. Some of the classic mouse behavioral tests, for example, the Morris water maze, we would never do that to a baby. We would never throw a baby in a pool and expect the baby to find a platform. So we have to use things like smell, which we know, for example, are very important in the developing human infant. We decided to compare the integrated transcriptomes of both humans and mice, and then do proof of principle experiments to evaluate candidate therapies. And in parallel, in work that I'm not going to present here, we are working with Tomo Terui here in Boston, who is studying human fetal MRIs to be able to determine endpoints that we can use when we start our human clinical trial. So the 3 most commonly used mouse models of Down syndrome are quite different from a cytogenetic basis. The most common and the best known one is the TS65DN, which has been in use for over 20 years. The challenge with human Down syndrome, human chromosome 21 does not have an exact copy in the mouse. It's primarily orthologous to parts of mouse chromosome 16, but there's a little bit of 17. So you see here this teal color is all 16, the brown color is 17, and the purple color is 10. So trisomy 21 in the TS65DN mouse is mainly part of 16, part of 17, and then there are genes that are not present in the human. And those are listed down here, and in particular, the one that I'm concerned about is ARID1B is triplicated, because that is a gene that is known to be involved in autism. So you're really not getting an exact phenocopy of the human condition. Now, we were very interested in TS1CJE. This is a translocation which has a large part of 16, but it also has some genes from 12, and it doesn't have as much of 16 as TS65DN. The advantage is the phenotype is a little bit milder, but the males are fertile. And that is very important to us because we want to use a wild-type female with an affected male, so that half of the pups will be affected. We will have a normal or typical mother fostering the pups, as opposed to a mother that may have developmental abnormalities. And then there's the duplicated 16, which actually has a duplication of most of chromosome 16 and none of the other chromosomes involved. In theory, that should be the perfect model. So we set out to compare them. This had never been done before. We compared the TS65DN, the TS1CGE, and the duplicated 16 versus their wild-type littermate controls. We looked at 3 points in their lifespan. We looked at embryonic day 15.5. We looked during the neonatal period up to 3 weeks, and then we looked at adult mice ages 6 to 8 months. We compared anatomy and growth. We compared the central nervous system histology, the brain transcriptome, and then we looked at animal behavior. This was work that was done in collaboration with Tarek Haydar's lab here at Boston University. This was the longest paper that I've ever been involved in. It was a 12,000-word paper. We're very grateful to Disease Models and Mechanisms because they accepted it and it has everything you ever want to know about these 3 models. And the reason why it's important is the models are completely different. It shouldn't be surprising because cytogenetically they are different, but they are treated very similarly in the Down syndrome literature. And we were very disappointed to find out that the so-called perfect model, this duplication of mouse chromosome 16, had no prenatal phenotype. Now that's very different, as I showed you in the first slide. The humans do have a prenatal phenotype. So this is not a good model for us to use. The TS65DN has the problem that the males are infertile, so we ended up, for now, using the TS1 CJE, but mouse models in Down syndrome research are still a very, very active area. But my word to you is be cautious because they are all very different from each other. So what we did was we took our cells from amniocytes, from human amniocytes that were confirmed to have trisomy 21, along with the amniotic fluid that contains cell-free RNA. We were also able to look at transcriptome that was publicly available in the literature from neurons and from fetal brain tissue. And we then used embryonic brains from our 3 mouse models of Down syndrome. And the reason why we didn't do RNA-seq and we used microarrays was because we were interested in using something called the connectivity map, which I'll explain in the next slide. But we were basically looking for differences between the affected tissue and the sibling controls or the wild-type controls. Now, why did we use the connectivity map? Well, that was developed as a way of repurposing FDA-approved drugs. Now remember, we're dealing with pregnancy. We don't have the capacity to develop new drugs and then test them to see if they're safe to use in pregnant women. So from the beginning, as a proof of principle, we wanted to use FDA-approved drugs. And the connectivity map takes data that are available from particular tissues in a state of interest, and then they perturb the cells or the tissue with 1,500 known FDA compounds. Each tissue has a signal, and then it is perturbed. If a signal is positive with the perturbogen, that means you are mimicking the phenotype of that condition. What we are looking for is we're looking for something that is highly negative to negate the condition, and that would be a candidate treatment. So we looked at all of these compounds, and one consistently rose to the top, and that was Apigenin. And here we're looking at 9 different cells or tissues, and we're looking for something that is the most consistently negative across the board. So Apigenin was clearly the first drug to try. We have some other ones on here that are actually negative, some that have been shown. For example, memantine is one of these drugs that has been tried in a human clinical trial and it didn't have any effect. So perhaps it's because it's not overall not that negative. That's potentially one explanation. And I put fluoxetine here because there is one human clinical trial going on for prenatal treatment of Down syndrome, giving mothers who do not have any psychiatric disease fluoxetine, which is Prozac, prenatally to treat the brains of infants with Down syndrome, and we're very interested in the results of this trial because we would predict that it wouldn't work. So we decided we were looking for something that was safe, that had a very negative profile on the connectivity map, and what came up, as I mentioned, is Apigenin. It's a naturally occurring flavonoid. It's otherwise known as 4'5'7"-trihydroxyflavone, and it is a potent antioxidant as well as anti-inflammatory compound, which are two of the areas that we're interested in. It's been shown to cross the blood-brain barrier in animals, and it promotes neurogenesis in the hippocampi of adult wild-type mice, and it improved learning and memory, which are key abnormalities in humans with Down syndrome. And it has already been used in humans. There are 5 studies that are available on clinicaltrials.gov. So I'm going to present to you right now the results of our proof of principle study, which is published. It's up on bioRxiv. It's undergoing review. And the hypothesis is that Apigenin will rescue the cellular and molecular deficits in human cells with trisomy 21 and in the TS1 CJE mouse model of Down syndrome, and that the prenatal treatment will improve postnatal neurocognitive outcomes in the TS1 CJE mouse, and that the post-treatment changes in gene expression and the dysregulated pathways will lead to testable secondary hypotheses regarding mechanisms of action. So the first thing was to decide what levels of the compound should we give. We exposed human amniocytes to different concentrations of Apigenin. We defined the optimal dose, and then were able to measure oxidative stress using the COMET assay. We measured antioxidant capacity, and we looked at global gene expression. And then we did our mouse models. We mated the C57 black 6 gams to TS1 CJE males. Half of them got a control diet, half of them got Apigenin. We used a powdered diet, so the diet was blinded to the investigators. And then we looked at gene expression in embryonic brain. In the neonates, we tested olfaction with the homing test, and then in the adults, we looked at open field and fear conditioning as behavioral tests. So the first thing we were able to show very conclusively is that Apigenin rescues oxidative stress and improves antioxidant capacity. So here we were looking at adding Apigenin to both euploid cells and cells from trisomy 21 fetuses. And you can see here that there is a lot of DNA in the tail, which is a harbinger of oxidative stress. So if you can see these slides, there is fluorescein-bound DNA that is in the tail of these cells as they're migrating across an electrophoretic field. When you incubate the cells with Apigenin, you lose that tail, you get a statistically significant improvement in oxidative stress. And similarly, the oxidative capacity improves with the same concentration of Apigenin. And so it, at least in vitro, appears to normalize this problem with oxidative stress. We observed that the prenatal treatment was safe. We did not increase birth defects. We did not increase loss of the pups. The transcriptome change was not as exciting as we hoped it would be. It would normalize the gene expression in many, but not all of the genes. Did worsen some of them, and I will show you how they mediate the changes through upstream regulators. There's a clear improvement in olfaction, and there's significant improvement in the fear conditioning test. However, we uncovered a major sex difference between males and females. So let me show you what that looks like. So here are the neonates. Here's wild type, untreated, TS1 CJE untreated. And what we're looking at here is how long it takes the pup to migrate or move itself to a piece of cotton that has been soaked with odor from the mother. and it takes a lot longer, so there's an abnormal sense of smell in the untreated TS1-CJE. But once you treat the TS1-CJE pups prenatally with Apigenin, you basically normalize the results of the homing test. And in the adult mice, again, and this, we now have broken this out, males and females, so there is a slight difference in males, with the males being worse as well, but it's much more significant in the adults. So here, instead of white, we have the wild types in black. I've lost my pointer, unfortunately. No, it doesn't show up there. So the untreated TS1-CJE, again, is orange, and then once you treat is in gray. Thank you. Once you treat in gray, you can see that if your wild type doesn't make that much difference, but there's a significant difference in the treated from untreated to treated. Looks great in the males, but gets slightly worse in the females. This is the open field test, so we're measuring exploration of the perimeter, and the females get more hyperactive. The fear conditioning test is testing the mouse's memory and ability to remember a shock that was given the day previously. Again, with the males are significantly worse, and the treated mice, so going from orange to red, are statistically significantly different. Doesn't really have much of an effect in the females. So this is very interesting because, as you know, until the relatively recent NIH rule that experiments had to be done in both male and female mice, all the literature is on male mice. And from the very beginning, we did all of our experiments on males and females. And it's very important because this may actually explain some of the difficulties in translation from mice, meaning male mice, to humans, which are both males and females. So here's dissecting, getting at some of the mechanisms. And the point of this slide is really to show you that human amniocytes and mouse embryonic forebrain are very, very similar. So in both of the situations, you see in the untreated state, you see genes and pathways that are associated with increased inflammation. When the cells are treated, that all decreases. There's decreased inflammation, and we see that from both the genes and the upstream regulators. The same thing occurs in the mouse. And the other issue is this decreased neurogenesis, and it's why the brains of humans with Down syndrome are significantly smaller than individuals with normal chromosomes. There is a lack of progression of cells through the cell cycle. Once you treat, you see a lot of cells moving towards metaphase. So it is definitely, we assume this is going to help with neurogenesis. You see the same thing in the mouse. And in addition, in the mouse, you see quite a bit of evidence showing that there is improvement in neurogenesis and genes that are associated with stem cell maintenance. Now, we think that this really requires a personalized approach, that not all humans are going to respond to the same drugs. And so here we have a series of different human amniocyte cultures, and this just shows you 2 different cultures' response to Apigenin. So we're looking at proliferation here. This particular cell culture is very responsive to Apigenin. This one is not. So we don't think there's going to be a one-size-fits-all approach, but the beauty is you're getting human fetal cells as a result of the amniocentesis. So as part of confirmation of the diagnosis, you would actually have access to that human fetus's cells, and you could design the therapy that's appropriate for that fetus. So in summary, analysis of the amniotic fluid transcriptome shows reproducible patterns for each genetic disorder that is consistent with its known pathophysiology. We've used the connectivity map to repurpose FDA-approved drugs and test treatment hypotheses. What I've shown you here is that our initial work with Apigenin shows a proof of principle for treating trisomy 21, but the same approach could be used for other genetic conditions such as Turner syndrome. However, sex as a biological variable must be considered. We think that at a minimum, Apigenin is achieving its therapeutic effects in trisomy 21 by reducing inflammation and advancing the cell cycle. And in vitro, it's rescuing redox homeostasis imbalance. And in the mouse model, it improves behaviors, but with sex-specific effects. So in closing, I just want to acknowledge the many people who have been involved in this work, and some of the comparison work that I mentioned to you is also in collaboration with Boston University. This is our laboratory at the Human Genome Institute, and some of our bioinformatics work has been done in the Netherlands, and some of the cells have been supplied by our collaborator at Women and Infants. So I'd like to thank you for your attention. I'm happy to take questions on this or on the strategic plan. Thank you.